Hello everyone, my name is Yaqing Guo from University of Hohenheim. It's my great honor to introduce our recently published work in iMeta to you. The topic is about genetic diversity and the community composition of Arbuscular mycorrhizal fungi associated with root and rhizosphere soil of the perennial plant Pueraria phaseoloides. As you can see from this picture, gold mining caused a huge loss of agricultural lands, especially in Ghana. Of course, urgent actions are needed to mitigate such ecosystem degradations. Plant AMF has been recognized as an efficient way. However, understanding the extent to which various factors modulate abundance, diversity, and selective root colonization of AMF is essential because actually many factors have been reported to shape AMF communities. For example, geographic locations, soil conditions, plant species, and so on. However, AMF diversity and communities in association with perennial plants in heavily degraded ecosystems remain largely unexplored. Then, in order to explore AMF diversity and compositions in such kind of degraded ecosystems, we select two different places in Ghana, as you can see from here. Then we follow these procedures to get our DNA from rhizospheres and the root. Then come to the results. As you can see from figure A, relative abundance in rhizosphere soil, Acaulospora is the dominant species. In root compartment, Rhizophagus as dominant species. Come to the AMF structures. As you can see from B, compartment is a significant factors to separate from the root and the rhizosphere structures, however, not locations. Then we also did area diversity analysis. As you can see from evenness and diversities, this rhizosphere soil had a higher evenness and diversities compared to the root compartment in both locations. Furthermore, the seek-to analyze showed the differences of amplicon sequence variants between the two plant compartments. As you can see from here, the rhizosphere soil compartment, here is the root compartment. Finally, we also did the co-occurrence network to identify keystone species in both compartments. Then we found that ASV 230, which is belonging to Acaulospora, as keystone species in rhizosphere soil compartment, and ASV 238 is a keystone species which is belonging to the Rhizophagus in root compartment. To conclude, we showed that the perennial plant Pueraria phaseoloides had a strong modulation effect on AMF communities. Secondly, irrespective of geographic location, community composition of AMF in rhizosphere soil differed from that of the root. Finally, it revealed two highly abundant AMF keystone species in rhizosphere soil, Acaulospora, and the roots Rhizophagus of Pueraria phaseoloides. Thank you very much for your attention. Please find more details in this link. Thanks a lot.